This is a recorded lecture for Chapter 6, Microbiology 230. I am Dr. McGraw. In this chapter, we're going to look at microbial metabolism. In other words, how we fuel the cell growth. How does that happen? This is kind of a continuation of what we saw in Chapter 4 earlier. So, a glimpse of history, if you will, as we look at this again. We see that biologists had noticed that in vats of grape juice and alcohol and CO2 are produced while yeast cells increase in number. But it wasn't really widely accepted as mocked by some, and in the 1850s, Louis Pasteur set out to prove that simplified setup, clear solution of sugar, ammonia, mineral salts, and trace elements, added a few yeast cells and they grew, sugar decreased, alcohol levels increased. So it strongly supported the idea, but Pasteur failed to extract something from inside the cells that would convert to sugar. In 1887, Eduard Bucher, a German scientist, showed that crushed yeast cells could convert sugar to ethanol and CO2. He was awarded the Nobel Prize in 1907 for that discovery. So look at microbial metabolism. All cells need to accomplish two fundamental tasks. They need to synthesize new parts, and that is cell walls, membranes, ribosomes, nucleic acids. They have to harvest energy to power reactions, and the sum total of chemical reactions is called metabolism. And that's that process. The implications of microbial metabolism include biofuels we can create, food production, an important laboratory, invaluable models for study, and unique pathways for potential drug targets. The principles of microbial metabolism we can separate metabolism in two parts: catabolism and anabolism. And I always try to remember these in kind of a funny way. So anabolism is think about anabolic steroids. That's what these sports stars take when they want to have bigger and better muscles. So they take anabolic steroids. So anabolism is taking smaller things like small atoms and small molecules and making larger molecules, if you will. And that's anabolism. So that's the process where we assemble subunits of macromolecules to create larger ones. Now, catabolism, I always remember by cannibalism. If you ever go to some place where they're cannibals, they take you apart bite by bite. They don't put you together. So that's what catabolism is. The process of degrading compounds from larger molecules down to smaller molecules or atoms. And so the cells, catabolism, the process the cells capture to make ATP, and then anabolism, we have ATP is required to drive the reactions. And these two processes are pretty well linked in the cell because one sort of drives the other, and vice versa. The energy is the capacity to do work, and there's two types of potential energy. There's potential energy, and that's stored energy. And then there's kinetic energy. And I like to explain potential kinetic this way. Potential energy is stored energy, and kinetic energy is energy that you use. For instance, when you drive your car, you have gas in the gas tank. That's your potential energy. But some of the gas in the car is moves to the engine, and in your internal combustion engine, it gets ignited, and it causes the engine to move and off the car to move. So that's the kinetic part of it. So potential is stored energy, like we see it in we see it in chemical bonds, a rock on the hill that's going to roll down, water behind a dam. If the dam breaks, the water would move down. That's all potential stored energy. Kinetic energy is energy of movement, like moving water. So it's this hole in the gate, if you will, that's not allow water to go through. That's kinetic energy. Energy in the universe cannot be created or destroyed, but it can be converted between forms, but not very efficiently. So we can create between different forms, but not in an efficient sort of way. So photosynthetic organisms, they harbor energy in sunlight. The power synthesis of organic compounds form CO2. They convert kinetic energy of photons to potential energy of chemical bonds, so they produce their own potential energy. Chemoorganotrophs, they obtain energy from organic compounds, which we talked about earlier. They depend on activities of photosynthetic organisms. They depend on the chemolipid autotrophs in some environments. So you see that all of these have a relationship in the chain of life, so to speak. Now, free energy is energy available to do work. For instance, energy released from chemical bond is broken. Now, we compare free energy reactants and products. We have ergonic reactions and exergonic reactions and intergonic reactions. An exergonic reaction is where energy is released, exergonic reaction. Intergonic reactions are where the reactions sort of save energy. So exergonic usually means a breaking of a bond, a chemical bond. Intergonic is the creating of a chemical bond. And a change in free energy is the same regardless of the number of steps involved. So we can convert glucose to CO2 and water, for instance. The cells use multiple steps for degrading compounds. Energy is released from exergonic reaction powers. and from exergonic reactions from the intergonic storage units, if you will. There's nice feedback inhibition of biochemical pathways. You can see here. You can see that if you get on your D2L and download the PowerPoint for Chapter 6, you can then watch that. You have to put it on slideshow, and then you can watch it. The only way to do it. I can't do it in this mechanism. Now, metabolic pathways are a series of chemical reactions that convert starting compound to end products. They have to be linear or branched, or they can even be cyclical, as a matter of fact. So we have a linear version, starting compound, intermediate, and finally the end product. Starting compound, in this case, a branched intermediate creates two other intermediates, which create two end products. And then circular, the starting compound here goes through a series and finally get an end product. So there are different pathways that do that process. The role of enzymes is they're biological catalysts, and that means that they speed up the process, and they cause it to require less energy to happen. And what's really neat is once it's done, the enzymes convert back to the form they were before, so they can do the whole process again. So biological catalysts speed up the conversion of the substrate, that's the thing they're working on, into the product. By lowering the activation energy, they're highly specific. They only work on one enzyme, works on one thing. In fact, they're named for that. So catalase, if you were oxidase, only works on oxygen, for instance. And so highly specific, one at each step. Reactions wouldn't occur without it, but they'd be extremely slow. And so you can see here, without the enzyme, you see that it takes a lot of energy, and it takes a while to do that. If you have the enzyme, it takes less energy to do that, and the reaction happens at a quicker pace. How enzymes work, that's also located in the slideshow on this PowerPoint. Now, the role of ATP, adenosine triphosphate, ATP, is really the energy currency of the cell. It's composed of a ribose unit, adenine unit, and three phosphate groups, if you will, adenosine triphosphate. The adenosine triphosphate, except for free energy, is the acceptor for free energy. So cells produce ATP by adding phosphate to ADP, and that connection of the two phosphates, the third phosphate and the second phosphate, creates a highly exergonic, highly energetic bond. And so when it's broken, it releases an extreme amount of energy. So reduce energy from ATP to yield ADP in that phosphate. The three processes that do that in a cell: there's substrate level phosphorylation, and that's an exergonic reaction. There's oxidate phosphorylation, and that's where oxygen is used to drive the force, protein motive force. That's the most efficient. And photophosphorylation, which uses sunlight to create the proton for the motive force. So that's the different mechanisms we have. The components of metabolic pathways, the role of the chemical energy source and the terminal electron acceptor. Some atoms and molecules are more electronegative than others, and so they have greater affinity for electrons. Energy is released when electrons move from a low affinity molecule to a high affinity molecule like glucose it breaks down to water. You can see the energy, there's the organic compounds, and you can see the terminal electron receptors over here. Now, the role of chemical energy source in the terminal electron acceptor allows more energy release when the difference in electron negativity is greater. So electron donor and there's electron receptor. Electron donor is the energy source. Electron acceptor is the terminal electron acceptor. So the electron donor releases electrons and the acceptor accepts those electrons. Now, prokaryotes are remarkably diverse in how they use the energy sources and terminal electron acceptors. So organic, inorganic compounds are used as an energy source, and O2 and other molecules are used as terminal electron acceptors. So electrons are removed through a series of oxidative reduction reactions, or what are called redox reactions. Substances that lose the electrons in this chemical reaction are called oxidized, and substances that gain electrons is called reduced. And I always like the term oil rig. Oxidation is loss, reduction is gain. It's just I don't use an acronyms much, but it's one that kind of comes to mind. Electron-proton pair or hydrogen actually moves from one to the other, and the dehydration is oxidation, and hydrogenation is reduction. So you see that movement of that one electron. The role of electron carriers: energy is harvested in a stepwise process. Electrons are transferred to electron carriers, which represents reducing power. Easily transfer electrons to chemicals with high affinity electrons, so they raise the energy level of the recipient molecule. Think of them as carriers of electrons and the electrons are the driving force for energy. So NAD, NADH, NADP, NADPH, and FAD and FADH2. These are electron carriers, if it were. And so they are energy. Think of them as gasoline. And the gasoline has different octanes, and so they have different octanes that help drive some sort of reaction downstream. This is a look at a mitochondria, and again, there's a nice video located in this PowerPoint. Now, precursor metabolites are intermediates of the catabolism that can be used in metabolism. So they can be used in the process. So they're not the end product, but they're byproducts that can be useful. They serve as carbon skeletons for building macromolecules. For example, pyruvate can be converted to amino acids, alanine, leucine, or valine, if you will. And it tells the precursor metabolite, the gateway where it's generated, and the biosynthetic role. You can see that there. For instance, recall that E. coli can grow glucose salts in the medium. It contains just glucose and inorganic salts. So glucose is the energy source, and glucose is the starting point for all cellular components. It includes proteins, lipids, carbohydrates, and nucleic acids. Some glucose molecules are completely oxidized for energy. Others are used as biosynthesis process we talked about. There are three, when you look at catabolism, there are three central metabolic pathways. That is, we oxidize glucose to CO2. The catabolic and precursor metabolites, as well as the reducing power, can be diverted for use in biosynthesis, and this is termed anabolic to reflect this dual role. There's glycolysis. In glycolysis, this splits the glucose into two pyruvate chemicals, and they generate a modest ATP reducing power, not very much, about 2 per cycle. Pentose phosphate pathway is a primary role in production precursor metabolites, and this is also a pretty modest production. Tricarboxylate acid cycle oxidizes pyruvates for glycolysis, and it generates reducing power precursor metabolites and ATP. So these are different mechanisms to make that happen. This is how glycolysis works. I'll let you play that video in your PowerPoint. Now, central metabolic pathways include glycolysis, pentose phosphate pathway, and tricarboxylate acid cycle. And the key outcomes are ATP, reducing power, and we're called precursor metabolites. We'll take a look at this as we go through. So this is the outside of the cell. It talks about the first step of that. Respiration transfers electrons from glucose to electron transport chain. Electron transport chain is where most ATP is created. It's the most optimal methodology. So the electron transport chain generates these proton motive forces, or free electrons, and they're harvested to take ADP and a phosphate and make ATP out of it via this oxidative phosphorylation. And it requires aerobic respiration, and the O2 is the terminal electron acceptor, and anaerobic respiration, a molecule other than O2 is a terminal electron acceptor. It also uses a modified version of the TCA cycle. So what we're seeing here is that this very efficient system that provides most of the ATP. Again, a great video you might want to watch that process. Another photosynthetic electron transport and ATP synthesis you can see in the cell. Again, another great video. Fermentation. If cells can't respire or go through respiration, they're running out of carriers available to accept the electrons, and glycolysis will stop. And so fermentation uses pyruvate, or derivative as a terminal electron acceptor, to regenerate NAD. So glycolysis that can then continue, and you see the metabolic processes. The uses an electron transport chain, yes or no, terminal electron acceptor, and then you see the ATP generated by substrate level phosphorylation, ATP generated by oxidative phosphorylation, and total ATP generating. See the differences here. Again, you can see that oxidative phosphorylation is by far the most efficient methodology. But enzymes are biological catalysts. And the name reflects function. Whatever they're working on ends in A's. So it has active sites in which substrates bind weakly. So it causes the enzyme shape to change slightly and induces a fit. The existing substrate bond is destabilized and new ones are formed, and the enzymes are highly specific for a given substrate. Enzymes are not used up, as it were. Here's a good look at that. There's two different views of this. So here we have a substrate. It attaches to the enzyme. The substrate is broken down, and the bonds are broken. And so this substrate can work in both directions. It can create bonds or break down bonds depending on the enzyme. Whatever the enzyme is based on the substrate. For instance, if this was protein, this would be proteinase. So A's is what it ends in. It breaks down protein. Or peptidase, if you will. Peptidase creates a different, if you will, breaks down protein bonds. So there's that mechanism there. You can also see that this enzyme, the substrate, that goes into this little locked area here, and it's then broken down. Same mechanism, just a different way to look at that. Now, cofactors also assist in some enzymes. So in other words, here's an enzyme, but it can't work on the substrate unless it has a cofactor. The cofactor is the limiting control. So cofactors can assist different enzymes, including magnesium, zinc, copper, and other trace elements can be that cofactor, allowing the substrate to connect the enzyme and be broken down. And there are lots of coenzymes, and again, they are limiting. They can be limiting factors. So in other words, this enzyme will not work unless the cofactor is present. So coenzymes are organic cofactors. They include electron carriers like FAD, NADH+, and ADHP+, and fewer types are needed. So it's derived from vitamins, and usually B vitamins are part of that cofactor. Now, there's environmental factors that influence enzyme activity. Enzymes have a very narrow range of optimal conditions as we're talking about the environment. The temperature, the pH, and the salt concentration can change the optimization or optimability of a given enzyme. So 10 degree centigrade doubles the speed of the enzymatic reaction up to this maximum temperature here. So even this temperature, if we keep increasing the temperature, the function will decrease, and the proteins will denature at higher temperatures. Low salt, neutral pH, which is around 7, is usually pretty optimal in this process. We also have what's called allosteric regulations, and the enzymes are actively controlled by binding to an allosteric site. So let's look at this. There's the enzyme, and there's the allosteric site. So the enzyme will attach to the substrate unless the allosteric inhibitor is here. Now there's two ways this can work. You can have an allosteric inhibitor or an allosteric accelerator. So let's see what that means. So we have an inhibitor here. It will block the enzyme from being broken down, or block the substrate from being broken down by the enzyme. And thus it only works when the inhibitor is not there. If this were an activator, that would mean the allosteric site would have to be in place. And if it wasn't in place, it would look like this. And it only when it was in place could the enzyme be broken down. So there's some that turn it off and some that turn it on. An allosteric inhibitor turns it off. An allosteric accelerator turns it on. And so it distorts the enzyme shape, prevents or enhances in binding. Regulatory molecules usually in product, and it creates this feedback mechanism. So one of the byproducts is this allosteric inhibitor that was created, and when that gets higher, it starts to come back and inhibit the production allows the enzyme to create the production or breakdown of that substrate. Now, enzyme inhibition is a site which inhibitor binds in terms of the type. For instance, it's a competitive inhibitor. So this is a PABA, which is a substrate to be broken down, and there's a competitive inhibitor. So if you have a lot of competitive inhibitor, it dilutes the substrate that the enzyme is trying to break down, and so it's less likely that the substrate will be broken down or reach the point, the site on the enzyme to be broken down. So it's a competitor, and it helps to minimize the breakdown speed of the enzyme. Now, non-competitive inhibitors are used to bind to a different site other than the active site. Like allosteric inhibitors are one example. The action is reversible. Some non-competitive inhibitors are not reversible. For instance, mercury oxidizes the SH group of amino acids. Cysteine converts to cysteine, converts to cysteine. The cysteine cannot form the important disulfide bond, and the enzyme changes shape and becomes non-functional. So enzymes can even, or some inhibitors can even do that. This is a good example of how the Krebs cycle works. I'll let you look at that on your slideshow. Now, we also have ATP. It's reducing ATP and reducing power with NADH, FADH2, and NADH-PH in the central metabolic pathways. We have precursor metabolites. The glucose molecules can have different fates and can be completely oxidized by CO2 for maximum ATP. So this is looking at that metabolic pathway we talked about. It can be siphoned off as precursor metabolites for use in biosynthesis. So we're talking about a cycle here to create that ATP. Just to look at that process again, you can see this within the PowerPoint under D2L. The central metabolic pathways starts with glycolysis. Glycolysis converts one glucose to two pyruvate molecules, and it yields two pyruvate molecules and two ATPs and two NADHs. And those NADHs are going to be used later in this process. They'll be accumulated later in the process. So investment is you have two phosphate groups are added. The glucose is split into these three carbon molecules, and three carbon molecules. That is the pyruvate. The payoff phase, three carbon molecules are converted to pyruvate, and it generates the 4 ATP. It does, but unfortunately it uses 2 ATP to do that, so the net is only 2 ATP. So in glycolysis, 4 ATP are created, but the glycolysis uses 2 ATP, 2 ATPs worth of energy. So the endpoint is it yields 2 ATPs, starts out with 4, uses 2 and gets 2, and then 2 NADHs. The pentose phosphate pathway also breaks down glucose. It's important in biosynthesis of precursor metabolites. The ribose phosphate or erythrose 4-phosphate also generates reducing power by NADPH. The yield is dependent upon the alternative pathway taken, if you will. The transition step, this is sometimes called the Krebs cycle. What happens is the pyruvate is changed, if you will, into coenzyme A. And coenzyme A, it then becomes the driving force of this Krebs cycle. Now, the Krebs cycle is a cycle, and I don't want you to memorize it, but it's a series of reductions or changes of one acid to another, and there's certain things given off, like, for instance, CO2 and ATP. And for every coenzyme A produced from one pyruvic acid molecule, it goes through the cycle. So each cycle creates 1 ATP. 2 cycles creates 2 ATP. And you can get the idea. It produces NADHs and FADH2s. And so you get that payoff of this. So every cycle, think of coenzyme A made from the pyruvic acid as a quarter that goes in a slot machine. It goes around the slot machine, and it kicks out 2 ATPs, and it kicks out 3 NADHs, and it kicks out 1 FADH, if you will. So it kind of produces that for you. So 2 of those would create 6 NADHs and 2 FADH2s. Now, the tricarboxylic acid cycle completes oxidation of glucose. It produces 2 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s. They use the term tricarboxylate acid. You'll see the term Krebs cycle often too. It has some precursor metabolite. That's the endpoint of this Krebs cycle. Again, I don't want you to memorize the enzymes. It's just not very helpful. I want you to know what's going in and what's coming out in glycolysis, and now in the TCA cycle or Krebs cycle. Now, respiration uses reducing power NADH and FADH2 generated by glycolysis, and that transition step in the TCA cycle to synthesize ATP. It's the big step. And that step is called electron transport chain, which uses redox reactions, a series of redox reactions like this cascade, to produce ATP. And it produces ATP at a fantastic rate, like about 34 ATP in bacteria and prokaryotes. The process proposed by the British scientist Peter Mitchell in 1961 initially was widely dismissed, and then Mitchell conducted years of self-funded research and received a Nobel Prize in 1978. And it's called the chemiosmotic theory, if you will. Electron transport chain is a membrane-embedded electron carriers. We see this cascade, if you will. They pass electrons sequentially from the NADH2 and the NADH plus and the FADH2 down this process of electrons, and you have a total of about 10 NADHs and about 2 FADHs, and you're able to pass those down. And prokaryotes, that's done inside a plasma membrane, and eukaryotes is done in the mitochondrial membrane. And energy is gradually released, and it releases coupled with the ejection of protons, and it causes, again, a massive production of ATP from ADP and phosphate. The components of electron transport, and most carriers are grouped into large protein complexes that serve as protein pumps, and there's 3 general groups: quinones, cytochromes, and flavoproteins. And the quinones are lipid-soluble molecules. They move freely. They can transfer electrons between the complexes. Cytochromes contain heme. The molecules with iron atom at the center, several types. And flavoproteins are proteins to which a flavin is attached. And FAD, other flavins synthesized to form riboflavin. So the point is that all of these are the steps in this gigantic cascade that creates these multiple ATPs. So NADH H plus goes in, FADH2 goes in, and that's the process. The general mechanism of proton ejection: some carriers accept only hydrogen atoms, proton electron pairs, others only electrons. So the spatial arrangement membrane shuttles the protons to outside the membrane. When the hydrogen carrier accepts the electron from the electron carrier, it picks up a proton from inside the cell or the myocardial complex in a eukaryotic cell. When the hydrogen carrier passes electrons to the electron carrier, protons are released to the outer cell or the intermembrane space of mitochondrial eukaryotic cells, and the net effect is the movement of protons across the membrane. And that's the driving force, the fuel, if you will, that causes the production of this turnstile, this pump that creates ATP. It establishes concentration gradient and is driven by the energy release during electron transfer. Now, electron transport chain of mitochondria, there's complex 1, 2, 3, and 4 that use different complexes, if you will, that cause that. I don't care if you remember this so much, as you just get an idea there's 4 groups. And the first one is NADH dehydrogenase complex. It accepts electrons for NADH and transfers it to ubiquinone, and it pumps 4 protons. Complex 2 is succinate dehydrogenase complex. It accepts electrons from the TCA cycle via FADH2. It's downstream of those carried by NADH, and it transfers electrons to the ubiquinone. Then complex 3 is cytochrome, a BC complex that accepts electrons from ubiquinone. The complexes 1 and 2. The 4 protons are pumped. Electrons transfer to cytochrome C, and cytochrome C oxidase complex is the one in level 4. And what you're seeing here is the 4 levels, but I want you to understand the electron transport. I want you to all go back to this 4-step process, in that this is basically a series of redox reactions, reduction oxidation reactions, that create a release, a movement of protons, free electrons, across to a pump system, either in the cell wall, in the cytoplasm wall membrane, if you will, in a prokaryote, or the mitochondria in a eukaryote, and that process of those flowing through caused a production of ATP from ADP and free phosphate. Now, so the general mechanisms of proton injection are that some carriers accept only hydrogen atoms. We said that. And other only electrons. We already talked about that during this process. So complex 1, 2, 3, and 4, we've talked about that. And this really looks at the electron transport chain of mitochondria. And I don't think this is helpful to look at this, but think of this as a pump. And the protons are pushed through the pump, and that pump turnstile spins, and it creates energy, it creates enough energy to attach a free phosphate to a diphosphate to create the triphosphate. That's a good way to experiment to say that. And often it's called a turnstile. You'll hear that term. Electron transport chain of prokaryotes has tremendous variation. Even a single species can have several alternative carriers. For instance, E. coli serves as an example of versatility of prokaryotes. As aerobic respiration E. coli, it can use 2 different NADH dehydrogenases, and the proton pump, equivalent to complex 1 of mitochondria. Succinate dehydrogenase, equivalent to complex 2 mitochondria, and produce several alternatives to optimally use different energy sources, including free hydrogen. The lack equivalence of complex 3 or catachrome C, so quinones shuttle the electrons directly to ubiquitinol oxidase, a terminal oxidoreductase, and the 2 versions are for high and low oxygen requirements. So even O2 can have variations based on that. Now, anaerobic respiration E. coli, it harvests less energy than aerobic respiration. So electron transport is very efficient when there's oxygen present, less efficient when there's no oxygen present. So lower electron affinities of the terminal electron acceptors and low oxygen content. Now, some components are different. They can synthesize terminal oxidoreductase that uses nitrate as a terminal electron acceptor that produces nitrite. E. coli can versus less toxic ammonia. Now, sulfate reducers use sulfate as a terminal electron acceptor, and produces the hydrogen sulfate as the end product. So that's the end product of their production, as it were, to create energy. Again, this looks at generating the proton motive force. I don't care if you know this process in the end. I just want you to know what we've talked about. Now, electron transport chain had a generation of proton motive force. So ATP synthase, harvesting the proton motive force to synthesize ATP. The required established gradient is released when the gradient is eased. ATP synthase allows protons to flow down the gradient cascade in a controlled manner. It uses energy as phosphate groups to ADP. So one ATP is formed from the entry of 3 plus protons. So calculating yields based on an experiment of rat mitochondria, 2.8 ATPs are made per electron pair for the NADHs, 1.8 ATPs are made per electron pair for the FADH2s. That we created in the other 2 cycles, the glycolysis and then the Krebs cycle. Now, you can do all sorts of things by calculating theoretical maximum yields of prokaryotes, talking about the ATPs you create. You do all that. That's not really helpful. It's just not, I just don't find that it's useful to do all this. The end point is that you get about 34 ATPs. It's slightly less than eukaryotic cells, about 32 ATPs. And so it's slightly less than that. So in prokaryotic cells, between the glycolysis transition step, TCA cycle, and the electron transport cycle, you get about 34 ATPs. In the eukaryotic cell, where it's inside the mitochondria, you actually only develop about 32, just so you know the difference. ATP yield of anaerobic respiration prokaryotes, the substrate level phosphorylation does yield you 4, but unfortunately, you only end up with 2. You use 2 of them during that process, so the gain is 2. Oxidative phosphorylation, you end up with a total of 22 in the process, so you get 34 total between that and TCA and all of that. So you get 38 sometimes in that force we talked about. This talks about the ATP yield of aerobic respiration prokaryotes. I don't care if you know all the steps, I just want you to know the parts we talked about. Now, let's talk about fermentation, because it's the alteration between what happens in some bacteria and some eukaryotic cells. Fermentation is used when respiration is not an option. E. coli is a facultative anaerobe, and aerobic respiration, anaerobic respiration, fermentation are all part of that process. Now, Staphylococcus pneumoniae lacks the electron transport chain, and so fermentation is its only option. ATP generating reactions are only those in glycolysis, and additional steps consume excess reducing power. So they regenerate NAD, if you will. So that's this process of fermentation. It really is creating end products that can be used, like NAD, farther down the line, for organisms that don't have a mechanism to produce their own NAD. The fermentation end products are varied. They're helpful identification, commercially useful, like ethanol, butyric acid, propionic acid, 2,3-butanediol, and mixed acids. So you can see the terms, the different out products that we have in the end. Now, catabolism of organic compounds, other than glucose and microbes, can be used. They can use a variety of compounds. They can excrete hydraulic enzymes, which transport subunits in the cell. They create fluid or appropriate precursor metabolites, which might be helpful. Like polysaccharides and disaccharides, amylases digest starch, cellulases digest cellulose. Disaccharides are hybridized to specific disaccharides, if you will. Disaccharadases. Now, lipids. Fats are hydrolyzed by lipase, and glycerol is converted to dihydroxyacetone phosphate, and it enters the glycolysis. Fatty acids are degraded by beta oxidation to enter the TCA cycle. And proteins are hydrolyzed by proteases. The inner groups are what deaminate is called, and those deaminations allow it to become a pyruvate-like molecule. So the carbon skeletons are converted to precursor molecules like pyruvate-like molecules. So they convert precursor metabolites to enter the appropriate pathway and on down to create the energy we talked about. Now, chemolithotropes, these are prokaryotes that are unique in the ability to use reduced inorganic compounds as sources of energy, for instance, hydrogen sulfide and ammonia. They're produced by anaerobic respiration from inorganic molecules like sulfate and nitrite, serving as terminal electron acceptors. And they're important examples of nutrient cycling. There are 4 general groups of these. There's hydrogen bacteria, sulfur bacteria, iron bacteria, and neutrophilic bacteria. Photosynthesis is another mechanism of creating energy. Plants, algae, and several groups of bacteria use that. And the general reaction is that light will create this energy in the end. X indicates that elements such as oxygen or sulfur as it's moving forward. That can be considered in 2 distinct stages: light reactions and light interdependent reactions. Light reactions or light-dependent reactions capture energy and convert it to ATP at that exact point. Light-independent reactions or dark reactions, they use ATP to synthesize organic compounds and involve carbon fixation. So it can be used in 2 different levels or different methodologies. There are many variations found in approaches, oxygenic or anoxygenic. This looks at the different mechanisms. Location of the photosystem includes type of photosystem, mechanisms for the generating the reducing power, and CO2 fixation. This gives you a nice look at that, and you'll want to look over that chart in your book. Photosynthesis captures radiant energy. The colors observed are those of wavelength reflection. So pigments are located in the protein complexes, the photosystems, within the membrane. So chlorophylls, like plants and algae and cyanobacteria, and bacterial chlorophylls, anoxygenic bacteria, absorb different wavelengths in chlorophylls. So sensory pigments absorb at additional wavelengths. So carotenoids, they are many photosynthetic prokaryotes and eukaryotes. The phycobilins, if you will, a cyanobacterium and red algae use those. And then antennae pigments form this complex, this funnel energy to reaction center pigments. So that's one of the mechanisms they use. Now, reaction center pigments, they donate excited electrons to electron transport chain. And chlorophyll, plants, algae, cyanobacterium, bacteria, chlorophylls, anoxygenic bacteria. So cyanobacteria as photosynthesis of the membrane of the stacked structures inside, and those cells inside the cell, and those organelles are called thylakoids. The plant algae and thylakoids in the stroma of the chloroplast, they have endosymbiotic theory explains that, as we said before in a previous lecture. Purple bacteria in the cytoplasmic membrane has extensive infoldings. Green bacteria, specialized chromosomes, are attached to cytoplasmic, or chlorosomes, excuse me, are attached to cytoplasmic membrane. So you see the different uses of that mechanism. The light-dependent reactions of the cyanobacteria and photosynthetic eukaryotes are two distinct systems called 1 and 2. The cyclic phosphorylation, photosystem 1 alone produces ATP. The reaction center chlorophyll is the terminal electron acceptor. Non-cyclic phosphorylation is used when cells need both ATP and reducing power. Electrons from the photosystem 2 drive the photophosphorylation and are then donated to the photosystem 1. The photosystem 2 replenishes electrons by splitting water, generates oxygen, processes oxygenic, and electrons are from the photosystem 1 are reduced to NADP plus to NADPH. This is a good cyclic and non-cyclic photophosphorylation. I'll let you look at that video when you download the PowerPoint. This looks at converting radiant energy to chemical energy. Basically, the excited chlorophyll chlorophyll in this will cause the reduction of NADPH into NADP, if you will, NADPH plus NADPH, if you will, downline, and that's one of the energy sources, if you will. This second version, photosynthesis 2, causes the breakdown of water, hydrogen. The hydrogen is used over on this side of the proton pump to create that process for the photosystem 1. The light-dependent reactions in anoxygenic photosynthetic bacteria each has a single photosystem. They cannot use water and electron donor, so they're anoxygenic. They use electron donors such as hydrogen gas, H2, hydrogen sulfide, H2S, or other organic compounds. For instance, purple bacteria use the photosystem similar to photosystem 2. The energy of electrons is insufficient to reduce NADH plus or NAD plus. Instead, they expand ATP to use a reverse electron transport. Green bacteria use the photosystem similar to photosystem 1. Electrons can generate the proton motive force or reduce NAD plus. Now, chemolithic autotrophs and photoautotrophs use CO2 to synthesize organic compounds or create what's called carbon fixation. In photosynthetic organisms, light-dependent reactions. They consume lots of ATP, reducing power. They reverse process of oxidizing compounds. The CO2 liberates a lot of energy, as it were. So the Calvin cycle is the most common use. It has 3 essential stages: incorporation of CO2 into organic compounds, reduction of the resulting molecule, and regeneration of the starting compound. There are 6 turns of the cycle, and that gain is 1 fructose 6-phosphate. It consumes 18 ATPs, 12 NADPHs per fructose molecule. So it's a reverse cycle, as it were. And this is how the Calvin system works. Again, a very nice video that you want to download. This is the carbon fixation process. I don't care if you know this. I don't think it's really as helpful. I just want you to know the previous slides. Now, in anabolic pathways synthesizing subunits from the precursor molecules, prokaryotes are remarkably similar in biosynthesis. That is, they synthesize subunits using central metabolic pathways. If the enzyme is lacking, the end product must be supplied. So fastidious bacteria require many growth factors. Lipid synthesis requires fatty acids and glycerol. Fatty acids will give you 2 carbon units, which are added to acyl group to form that acyl coenzyme A. So that what happens is the 2 carbon units, which are the pyruvic acids, are attached to the acyl group, which comes from the fat, and that creates acyl coenzyme A, which is going to drive that Krebs cycle or the TCA cycle. Glycerol, dihydroxyacetone phosphate from glycolysis. That's the other output of lipid, if you will. Nucleotide synthesis, DNA and RNA initially synthesizes ribonucleotides. The purines, the atoms added to ribose 5-phosphate, they form a ring. The pyrimidines, ring made and then attached to the ribose 5-phosphate. They can be converted to other nucleobases of the same type, if you will. Amino acid synthesis is a process of synthesizing of glutamate, provides mechanism for incorporation of nitrogen into organic material. Ammonium, NH4, commonly used to be a glutamate synthesis, and transaminization can generate other amino acids. So that's the process we have here to create those outputs. Amino acid synthesis of aromatic amino acids, carefully regulated branch points. The tryptophan is a feedback inhibitor of enzyme that directs branch to a synthesis. The pathway instead leads to tyrosine and phenylalanine. And tyrosine and phenylalanine likewise inhibit first enzyme of the branch leading to their synthesis. The 3 amino acids also regulate formation of the original 7-carbon compound doing 3 enzymes. You can see it here. There's the glycolysis from the one way, the pentase pathway the other way, and pentase phosphate pathway. You get a 7-carbon compound. Hits a branch point. Carbon A, carbon B. Compound A, compound B. Compound A gets a branch point 2. You get phenylalanine and tyrosine. And compound B, you get tryptophan. And this looks at that, just like subunits for precursor molecules, and that's what this really looking at. So what we've done here is we looked at the microbial metabolism and the fueling of cell growth, and we'll be moving on next to chapter 7 in the next recording, where it's the blueprint of life from DNA to protein, and that'll be an important step. So we finished this chapter. Have a good day.